everybody, and welcome to episode 72 of All About African Violets. All About African Violets is musically sponsored by Ted Yoder. You can hear and purchase his great music on his website at tedyoder.com. It is also available from iTunes. Hi everybody, come on into my sunroom. Not sunny, late in the day, and also a little early this week. I'm recording a little early because I'm going to be traveling again this weekend, and so I wanted to get everything ready to share with you for Sunday. And I have a very special treat for you today. This episode is a little bit longer than normal, but I have my last interview that I did this summer with Dr. Jeff Smith. And this time, it's all about the African violet species plants. And he is just so interesting. I can pretty much guarantee that you will learn something that you did not know before in this podcast this week. And the interview is more than 50 minutes long, so I'm making a couple changes because it is long. I just didn't want to split it because it's so interesting, and you just get into the flow of the conversation, and I didn't want to break it up into two episodes. So I'm going to forego the look at what's on the stands this week because I am recording early in the week. It does not look much different than it did when you saw it last week. So we're going to skip that this week, and really right now let's go to tips and treasures. I'm going to save I've got my African Violet magazine all marked of the things I wanted to share with you, but I'm going to save it for next week. So without further ado, let's go back to Muncie, Indiana, back to the summer when the days were warm, and this past summer when I went to interview Dr. Jeff Smith, and let's learn about the African violet species. I'll be back on the other side. Hi everybody, I'm here with Dr. Jeff Smith, and today we are going to talk about species. I love growing them.
Good.
I love growing. I've gotten some from you before. All the ones, unfortunately, that I got from you were in the collection when I lost it last year. So everything I had the Pangani Falls. I had a House of Amani, which I think I did pick up another one. I have one now that I'm growing. But I had quite a few from you that were so interesting, and I was so sorry to lose them. I was like, oh no. Yeah. But you have, you are known far and wide in the African violet world as being a real expert in the species plants. And we've heard you talk about your violets and how you kind of got started there, and the whole scientific piece of that, and some of the hybridizing and the things you've done. Is that how the species came to be for you to be important?
Well, the species kind of grew out of sort of a side interest. Okay. I was looking for some other projects to do some research on. And one of the things that I was curious about was whether, when we were working on the flower pigments and the chemical identity, did all of the species have the same pigment? Or was it different from one species to another?
Well, because they all have that bluish.
They all have that
lavender,
blue,
purple.
but there's different shades of it. So is it all the same thing, or is it a possibility of something different? Okay. So that was sort of the original question. And then the way I got into growing the plants or growing the species is actually out of a AVSA research grant, which allowed me to purchase a whole set of the species plants from John and Barbara Cook at Cape Cod Violet Tree, who was one of the primary sources for species plants for quite a number of years. And got that, and when I looked at them, when I unpacked them, it was like, oh wow, this is cool. Oh wow, this is cool too. Oh wow, look at the foliage on this one. This one's really cool. So one of the things that anybody who grows the species will tell you is that there's not so much a diversity in the flower color, because there isn't. Everything is a shade of blue, one or another. But when you think about, you've got miniatures, you've got trailers, you've got rosette plants, You've got large trailers, you've got shiny leaves, you've got very furry leaves. It's the difference in the species, or the attraction to me, is just the sheer amount of diversity that you have in the plants themselves, in the plant types, not so much the flowers, but the plant types. You've got leaves that are perfectly circular, very, very shiny, and so forth. And then when I started getting into the African violet articles and reading up on the species and their history and so forth, various questions came to light. And the question that really came out was, well, actually, how many species are there? That's been a real overriding question, in particular the last couple of years. But the interest sort of came out of the diversity within the groupings and all these different plant types and things like that. I actually had a student take on a science fair project under my guidance where he did a study that we called a numerical taxonomy, where he took the leaf sizes, length, width, flower sizes, number of hairs. Turns out there's more than one kind of hair on the African violet leaves.
Oh, wow.
And they're in different numbers and different ratios. So he took all of those kinds of things and plugged it into a statistical program and saw how the plants sorted out, and it got at least an initial suggestion of which species were more closely related to other species and things along that line.
Oh, wow.
Because one of the questions I had at the time is that you had Magingensis minima and Magingensis occidentalis, and they were all put into the same thing. They were all considered to be the same species. And I'm looking at these and I'm going, these don't even look alike. How can they be the same species?
Okay.
And I started doing some correspondence with Dr. Bill Burt, who was the person at Kew who had identified most of the species, at least quite a number of them originally, and raised some questions with him. And he goes, well, it's a little hard to say. We've only got like one plant from this locality, and it's different, so we named it, but I don't know if it's really an abnormality, or I don't know what all the other plants in the area look like. And that started to raise some questions in my mind, because many of the species plants that we had, particularly in the 50s and 60s, were single collection plants.
What do you mean when you say a single collection plant?
If you were to go out into a forest like here in Indiana, and I was to take a sample off of a sugar maple tree, okay, I would know that that was a genetically unique individual, and it may or may not be the same as any other sugar maple tree that grows in the state of Indiana or in your state of Illinois or any place else in the country. The African violets that we had here in the United States in the 50s and 60s were all descendants from one plant that had been collected in Africa, brought back over one single plant.
No, wait, I thought there was like the species in this, so there was more than one.
Well, there is more than one type.
But I mean, but all that we have now.
Well, what we
Or
would
initially,
have, what we had
1960s.
initially, was that when I would say, like, I have a plant of Velutina. That's a very popular one that people grow for show.
I can't grow.
Okay.
I've lost
okay.
countless of them. It's just
All right.
so irritating.
But what we tend to forget is that that plant that we call Velutina is just the descendant from leaf cuttings of one individual that was collected in the wild. It would be equivalent to me growing one sugar maple tree and passing it around all over the place, and everybody knows the sugar maple from that one specimen that we were growing in our area.
Okay,
So it's a
okay.
single collection plant. What we didn't appreciate is that in the sugar maples, if you start looking, you'll find little small differences from plant to plant to plant to plant to plant. Well, we didn't collect all of those. What was brought over from Africa, at least particularly in the 50s and 60s, was a single plant. It was all they got out. And so when we look at Velutina or those kinds of things, they're all leaf cutting descendants from a single individual plant that was brought over. We didn't have the whole population.
I had no
We
idea.
had no idea what the population of Velutina looks like, because we've only got one representative of it. So it's kind of like an animal example would be, okay, let's go out in the street and grab a dog. And that's what we know all dogs to look like, is that one dog that we grabbed off the street. And that's what we had with the species in African violets, is we grabbed one plant, we brought it back over, and that's what we thought everything of that plant looked like. And we didn't catch all of the little variations from individual to individual to individual within the population.
So is that how, this reclassification came about, or am I
It
jumping?
was.
Is that part of that with all the changes?
It's a part of the story.
Okay,
It's
so
a part. It's a part of the. It's a
don't
part
jump
of the
ahead,
story.
Andy. Don't jump ahead.
No, that's fine. No, it's very much a part of the story. I didn't get to appreciate until really much later on in my studies that these things were all single individuals. When I talked with John and Barbara and said, okay, where did you get your plants from?
Mm-hmm.
Well, they got them from Linden Lyon. Well, where did Linden get them from?
So that was the Cooks, and then
They went back to Linden Lyon.
Okay.
So by the time I got interested in this, Linden had passed. So I talked to Paul Serrano, his grandson, and said, do you remember anything about your grandfather having the species plants? He goes, yeah, Grandpa collected them, and we kind of had them thrown underneath the shelves, and he did do some hybridizing with them. This is where the trailers came out of originally, was out of the species plants.
Okay.
And then, but he just got them from people here and there and so forth. So I don't know where some of these actually got into the United States and where Linden got them from.
Okay.
But John and Barbara were the ones who really kind of codified it in terms of putting them all in one location.
Oh, wow. Okay.
And had them available, for where people could get a hold of at sale or whatever. Although there was some trading between individuals and that. The society actually set up a collection of species at Cornell University in what was called the Bailey Eye Garden. No, it wasn't the right name. Anyway, but most of those plants are now gone. And I've checked with Cornell, and they have none of them anymore. They've lost everything.
What a shame.
I still have one plant that has the Cornell number designation on it, but I don't know where it was collected from originally.
Okay.
That's where a lot of the things, we've lost the history of where in Africa specifically it was collected. We hear about the Diplotricha Parker, Diplotricha punders, those kinds of plants. Well, those names are after the collector.
Oh,
Parker
okay.
and Punter were the collector,
Okay.
who brought them over or sent them over to people here in the United States. And so when you talk about like Parker 0, Parker 7, Parker 8, or Punter, is it the other way around? Anyway, but those designations are all collectors' numbers,
Okay.
where they had a population and they'd sent 2 or 3 plants over. So they designated them by the number. The Grandifolia 299 and Grandifolia 237.
I have 237.
Okay, those are collectors' numbers. That's
Okay.
where those numbers come from. When collectors do a plant, they will put their number on it. This was the number 237th plant that I've collected and put and preserved and brought back.
Oh, that's interesting.
So that's where those numbers have come from. They just aren't arbitrary, numbers.
I never knew that. Did you guys know that? I never knew that.
That's where those numbers came from.
I just, I didn't know. I knew it's Grandifolia 237.
So why 237?
Yeah.
Well, that collector, that was his 237th plant that he had collected and put into
And that
the
one seems
reservation.
to have been the best one. It seems to be one of the ones you see all the time.
Yeah, it's one of the ones that does very, very well.
Okay.
So
I get it.
so you
Oh,
get
that's
these
so
you
cool.
get these little bits of history. All
We're
over.
geeks.
Yeah, we're geeks.
We're geeks.
So you
We
get,
love
you get
this
this little
stuff.
get this little bit of history. So then back, it's probably been 15 to 20 years ago now. I had some correspondence with some folks at University of Iowa. And, no, I take that back. Iowa State University. I got the wrong one there. Iowa State University, the Ag School for Iowa, at Ames.
I was just
At
going to say,
Ames.
isn't that the one
Yeah,
that
it's
Ames?
the one at Ames. Okay.
Okay.
They had a doctoral student who was wanting to do some DNA work on the species, and they had contacted some people in Tanzania and asked for plant material, and they actually got a shipment out of the country. How they managed to do that these days, I have no idea, because I cannot duplicate that. But they got a shipment out of the country, and these were plants that had been collected by a lady by the name of Silva Mather. You may have heard of the Mather
Mather, yes.
Mather plants.
Yes.
Okay, she lived in Nairobi, Kenya, and was interested in collecting the wild species violets and maintaining a collection of them. Well, she was killed in a traffic accident in 1992, I think it was. And another botanist in Kenya, who they had shared material for, a lady by the name of Ann Robertson, collected a bunch of Silva's plants, shipped them over to Iowa State, and then also to Kew. Well, and she went through and cataloged all the plants and had some notes about where it was collected or where Silva got it from, and these kinds of things, and had Silva's numbers on the plants or her abbreviations on the plants. So, for example, the plant that I'll show on the species tour, that's Chimensis EE. Well, that is a Silva Mather plant, and when Ann Robertson cataloged it, it was on the list, and she went like an alphabetical A, B, C, D, E, and then on the collection list, it was listed as EE. So I've tried to keep the labels that Ann and Silva had with the plants for identity purposes. So that's where you end up with some of these really strange numbers on plants, like Matha number 21 and Matha number 18, and things along that line. The plant that we call St. Paulia Robertson is an Ann Robertson collection from Kenya. It's a Rupicola plant clone. So we had these plants get in. Well, that like doubled all the available species that were here in the United States when I got a hold of the plants from Iowa State. They had sent me leaf cuttings, and I was able to propagate a number of the things that way. And then later on, I found out that they were giving up the collection because the student had left the university, was not doing the work, and they really had no interest in keeping them going. And I said, can I come out and have them?
Car ride?
Car ride, yes. Can I
ride.
come out and get them? And I got a little travel grant from the Indiana Academy of Science that gave me money to gasoline, drove out.
Just to get there.
Just to get there, drove out there. Got plants and collections from everything that they had, got a set of their records, and so forth. And they had a number of plants that were labeled as Uppsala, and at the time I thought it was St. Iverson because it was St. Iverson, and I thought it was a location name of St. Iverson. Come to find out that I had canonized somebody. It's actually Steve T. Iverson.
Oh.
And so it was the abbreviations on here. But it was a Swedish botanist who had done some collecting, and they had gotten some of his plant material. So now we have some plants from Uppsala, have some of the Iverson plants, and so that now increased the number of species things. And so I'm looking at all of these things, and I'm going, okay, some of these look really, really similar. So they're probably the same species, but other things, I don't know where to put some of this stuff. And other things are just plain weird. So that raised the question about how many species are there really with all of this. Well, to jump forward a few years, there was some DNA studies done by several people. Some folks at Kew, Michael Moeller was one. And I had actually sent him some plant material out of the collections here for them to include in their study. And a lady by the name of Charlotte Lindquist did some collecting in Africa and was doing some DNA work. And it was suggesting that the African violets are a very recent group of plants evolutionarily, and that they are very closely related. In fact, a bunch of them you couldn't even tell apart at the DNA level.
Wow.
Which suggests that they're very, very recent splits off of each other. And that raises the question, well, are they separate species or are they not? Because you tend to think of species being different genetically, and it turned out that the differences were very superficial. You couldn't really tell them apart at all. And then we found out that they're actually very closely related to Streptocarpus, the African primroses.
Oh.
In fact, the suggestions now is that the violets are a split-off of the African primrose group.
Really?
Yeah, they're that closely related, and it happened like within the last 10,000 years.
Wow.
Very, very recent split off. So that was making some suggestions of reorganizing what was species and everything else. Well, what really caused the fire was when a fellow by the name of Ian Darbyshire not sure I got the name right, did a publication that basically took all of our species and lumped them into 6 things. Just merged everything together into 6. And we looked at that, and I got asked by the society to take a look at his credentials, for one,
Sure.
and look at his work, and said, yeah, there's a lot of this that I can agree with, but I don't agree with parts of it. And then ultimately worked with Bill Price, Barbara Piershing, and another plant taxonomist by the name of James Smith. So we get some confusion between Jeff Smith and Jim Smith. He's at University of Idaho. And in consultation with him, we came up with the reorganized classification scheme that the Society uses now. So it basically recognizes 9 species. Some of them are the same as we had historically, like Schimmensis, Grotziana. Some of those we've kept separate. Other things got really lumped together, like Ionantha got lumped into a whole batch of stuff, got lumped into there. But we didn't want to lose the identification of these different genetic types. So we suggested using a designation with a clone name. So we would have like St. Paulia Ionantha, Variety Ionantha, Clone Ionantha, or Clone House of Amani, or Clone Uppsala 3091, these kinds of designations, which is more technical than most of us really want to worry about.
No, and we have talked a little bit about this on the podcast before, because this came up, and with these big long names, and they figured, how did we get that? And now when I look in First Class 2, I thought I put it in the right way, but now it looks like I can't tell which one it is, except you can once you're in there. And Joe explained that for us, and I knew we
and Joe,
and I was hoping
Joe
we would
worked
talk
with
about
us.
it. Yeah.
Joe worked with us a great deal on trying to reorganize First Class to reflect the new naming system. The biggest thing is to recognize is that there's really only 9 species that we recognize now. Ionantha does have 2 varieties underneath of it. Now, a variety is a natural occurring variation. It's not a man-made created thing like a clone or a cultivar. A man-created plant we would call a cultivar. So all of our man-created hybrids are cultivars, and that's what First Class lists. Taxonomists use the name variety when you've got a natural variation that you can tell apart. So under Ionantha, we have variety Ionantha, and we have variety Diplotricha, which means 2 kinds of hairs. And then underneath that, we've got all these different clones that fit into it. So really what we could have done was lumped everything in, but we know that like Grandifolia 239 and Grandifolia, or 237 and 289 look differently. And we were unhappy with the idea of making it all one name.
Yeah,
when they obviously look different. So the only way we could kind of figure out to keep some of those differences recognizable was to throw them in under the same species, which is what Devonshire. Anyway, what his work suggested needed to be done. But now attend on to the end of the label a clone designation. So we would have, I think actually Grandifolia is now a subspecies under Ionantha. And then we would put like clone 237 or clone 289. And we could tell them apart that way. Now for most of us, we can go straight to the clone name and tell it apart. But when it comes to show and they put the formal label in there, they're supposed to put all the things, and we suggested a numerical code where you could put 5A clone this, and then 5 is species 5, B is Diplotricha. We had a numbering system that would allow you to abbreviate it down, to shorten labels down to something like this. So instead of having species Ionantha, variety Ionantha, clone Ionantha.
You should have heard me reading that one.
These names, which are like yay long, and writing them on the pots. We could abbreviate it down, and really, if you just go with the last clone name, you'll be good, because you can always work backwards from that to everything that was preceding it in the formal label.
Okay.
So we've had that. Now we've had some other changes that have been suggested. Bill Price has brought back some additional plants from Uppsala that never did make it to the United States.
Oh, wow.
Pangani Falls is one of those. The Confusa hybrid that I showed on the tour is one of those ones. So we've got some things there that probably fits in the existing scheme. There was a publication out of Kew 2 years ago that recognized 2 brand new species, but neither of which are available here in the United States. We can't get them. Can't get them here. And I think they're probably going to fit in with some of the existing things, but nobody's done the DNA work in order to look at it. Now, the one thing that just really upset the apple cart within the last year is I mentioned that the violets look to be a subshoot off of the Streptocarpus, the African primroses. Well, in straight taxonomy fashion, if you're descended from the same ancestor, you should all be lumped together under that same name. So there was a fellow out of Iceland who basically said St. Paulia is no longer a valid name. We're going to lump them all underneath African primroses.
I'm sure lots of hair was standing on end.
Oh,
that one.
gosh.
Woo!
Okay, yeah. So in which case it affected the society, because the society has the patent, if you like, to name all of the St. Paulia cultivars.
Right.
Okay, the Gesneriad Society has the patent for naming the cultivars of any other Gesneriad except St. Paulia. So the question that I raised to the botanical taxonomy committees were, if this document holds and St. Paulia is now thrown underneath Streptocarpus, then who's got the naming rights?
Yeah.
Which way, what do we do? What do we do with this? And the answer that we got back is that let's take a wait and see attitude on whether people really believe this lumping into Streptocarpus. So for right now, we'll let the AVSA maintain the naming of the African violet cultivars like you've had. We'll let the Gesneriad Society maintain the naming of all the Gesneriad hybrids. We'll leave it like it was, unless something further comes down the road and shows that it should be different.
Okay.
Now, the latest DNA work is suggesting that Streptocarpus is probably too inclusive a group, and it probably should be split apart in a couple of subdivisions. If they do that, then I think what we're going to see is St. Paulia is going to be one of the groups that immediately goes off on its own.
Okay.
Because it's unique enough that I think it will stand alone as its own genus, and then our current classification scheme will remain intact.
Okay.
Nothing will change.
That would be good.
Which would be good. I'm hoping not have
really
to redo
good.
everything.
Holy mackerel.
Redo everything. But so from where we had this handful of species that everybody kept describing every time they found something new, and it looked a little different, they put a new name on it. That's what Bill Burt was doing in the 50s and 60s. We've kind of come full circle to recognize that the violets are all extremely closely related to each other.
Okay.
And there's probably only a handful of actually true species. But we've got a great deal of habitat destruction now going on in Kenya and Tanzania. So we may never really know on some of these, because the habitat's being fragmented up. We may never really know how some of these things related to each other, and we'll end up with some kind of classification scheme. And what we do for AVSA will be hopefully in line with what the plant specialists do, but we want it to be reflective. But we've got our own means and needs, like for show purposes, when we do the things.
Well, this is fascinating. I mean, it really is.
Probably way more history and so forth that people wanted to
I think it's
get into.
awesome. I'm like, want to take some home.
Yeah. Well, then we can arrange that.
Oh, I know.
We'll go downstairs and
Don't
do
limit
that.
my collection, but maybe we will have to go downstairs.
Well, one of the reasons that I maintain as many species as I do is to be a living history collection.
Yeah.
To be a living reservoir, because we never know exactly what we're going to need from the gene pool.
And when.
And if we don't maintain them, they'll be gone, because some of these plants are gone from the wild, at least near as I can tell from the reports that I get. And some of the ones still exist, but we can't get them out of the country and get them into cultivation. There's reasons for all of that, which I won't go into here. But the main reason I maintain as many of the species clones as I do is to try to be a living reservoir, a living collection. And so, then my collection's too big, and and I suffer for all of that. But that's one of my main goals with trying to keep the species going.
They are fascinating. And when you gave us the tour of your growing area, you referred to one that it was down on the bottom shelf and it was really big, and I think you called it a tetraploid. Could you explain what that is?
Tetraploids mean that it has twice the number of chromosomes that it's supposed to have.
Is that like the old Supreme Violets?
Yes, it's like the old Supremes and DuPonts.
Okay.
Same idea. So
how does that happen?
Usually by accident. You can cause it to happen if you treat the plants with colchicine. It's a chemical that'll interrupt the cell division.
Give you that for gout.
Yes, they do.
I know that.
They give you colchicine for gout. That chemical can be used to cause plants to double their chromosome sets. And so whether that one was an accident or whether it was done deliberately, I don't know. I don't know the source of that one. It's one of the ones the Cooks had. They got from Linden. Nobody knows exactly where it appeared.
Because I'd never seen it before.
Oh, okay. But it's
really
a monster.
amazing.
It's a monster.
It's so cool.
It's just a monster of a plant in terms of size. Tetraploids, like the old Supremes, are slower growing. They don't bloom as frequently, but when they do bloom, they've got huge flowers, huge leaves, but the foliage is very brittle, and they're very difficult to do any shaping. That was one of the complaints on it, and this one is more flexible than some. That's why that one Confusa plant, I suspect, might be a tetraploid, because you just pick it up and the leaves break.
Snap.
They just shatter.
Okay. Oh, wow.
So it's very brittle.
Probably why we don't see a lot of the old Supreme and those violets any longer.
Yeah, I think so.
I have another question, because you said, and correct me if I misheard you, that the trailers came from certain species.
Yes.
You talked about that. Now, I mentioned this, I was doing some reading. I have Francine Pilon's book, because she specializes in the trailers, and she was talking about some of the original trailers and how they, in the 50s, and how they were kind of ignored initially. And I have one, I got at National. I have Tinari's Royal Blue trailer.
Oh, wow.
And I'm so excited to have it. And I'm like, oh, I hope that it's going to be okay. But as we were looking downstairs at the plants, I can kind of see in some of your trailers that you did, the yesterdays that had species, in their heritage, the shape of the leaves, it's similar. They're not as they're shinier on the Tinari one, but I'm very interested in it. Can you speak to that a little bit?
What I've read, and this is going back to the old African Violet magazines and a little interviewing with the Tinaris when they were still alive, is that they had the idea for developing African violets that would cascade over hanging baskets and add that to the commercial world, essentially, as hanging pots, kind of thing. They started off with a species called Grodii, which is one of the trailers, but it's a standard. So their first trailers that they developed were standard trailers, and they were very coarse and huge and just all over the place. And the species are not known for their floriferiveness. So these didn't have that many flowers on it. And unfortunately, they came out with this in 1954 or 54, I believe it was. Thereabouts.
I'm thinking 54. I'm trying to think
54.
I'm remembering.
But it was the same year that Linden Lyon successfully came out with double pinks.
That's what she said in the
Yes.
book. That's what Francine said in the book.
And the double pinks had been a long-sought goal, and they didn't realize that double is a dominant and pink is a recessive. So in order to get double pinks, you would cross the double blue with pink. Your F1 hybrids would be all double blues, but then you would have to breed together or cross back to a pink to get the pink gene back out. So, and that year at the National, when the Tenaries introduced the first trailers, also was the year Linden Lion introduced the double pinks, and the double pinks had everybody gaga, and the trailers got no attention. Just got no attention. Now, Linden Lion went back later and used a different species. He used the Magangensis minima, a much more compact species, and created some of the first mini and semi-mini trailers.
Okay.
And those caught on. And this was, I think, into the 60s. It was almost a 10 or 15 year span later. And that then became the basis of many of the other trailers that we have now. They're descendants out of the Magangensis minima rather than the larger growing Grodii ones. What I've attempted to do with the Yesterday series is to kind of mix and match some trailing things together. One of the things I've used fairly successfully is a Mather trailer called Mather 21. And it has long pendulous trails just all over the place. But it passes that trailing trait on in the first generation.
Oh.
So I can cross it onto about anything, and I can get compact trailers. It's like the long compacts down, and I can get that in the first generation. Yesterday's Child has 3 different trailer species in its ancestry. It actually has Velutina in its ancestry, but that one's not a trailer. No, so okay, it has Velutina, Magangensis, and I'd have to look up the third one now, but it was, trying to combine some things together. You mentioned growing Yesterday's Valentine.
I have it. It's just getting ready to bloom.
Okay.
It's not too big yet. It's about like that.
Yeah, it'll get bigger. But it has Velutina in its ancestry, so it's got the foliage. Well, it's got the Velutina style foliage, and that, but on a trailing style plant. And that was the goal, was to try to get that combined.
I do not know what I do wrong with Velutina. I've grown it in the basement where it's cool. I've grown it upstairs where it's warm. I've tried it everywhere, and it just doesn't grow for me.
It's temperature tolerant, but it does not like wet feet. It's one that will not take wicking unless you have extremely porous soil mix.
Maybe that's it.
Because I've found that it is very intolerant of, when we say wet feet, it does not
Wet
like
all
to
the
be in
time,
standing
yeah.
water. It does not like to be wet all the time. Now, it does like being damp, but, to be soggy, absolutely not. It'll root rot faster
That's
and all
probably
get
what
out.
I did.
So that one is very temperamental, not so much on temperature, but it's much less tolerant of wet soil conditions than most of the other things. That's one of the other fascinating things about the trailers. Almost all of them in the wild grow on rock outcrops in, like this much soil, on logs, or on moss-covered boulders. They are in extremely shallow soil, which is why our soil mixes need to be so light and airy and draining.
So even more lighter for a trailer, you would say, than a regular?
My favorite for growing any trailer is I grow it in the little saucer pots, the pan pots, and I actually fill the bottom half with almost straight perlite.
Yeah.
And then put my soil mix on top of that.
I do that.
And grow it as shallowly as possible.
But do you cut your soil mix to make your soil mix even lighter?
Yeah, I will at times.
Okay, I'm going to try that.
I will at times. The great big giant trailer that I was showing you.
The one in the dining room?
The one in the dining room. It's probably, the pot is only about that deep, and probably half of it is perlite.
Okay.
I mean, the actual roots are going down through that. But trailers in particular are out of an ancestry of growing over rocks, growing over moss-covered boulders. And in fact, all the species plants are rock growers. So they, that's why it's deft to have them in heavy soil. Any violet just can't take heavy soil. Trailers may be a little bit more sensitive than some.
Okay.
You were talking about my growing conditions a little bit earlier. I'm surprised you didn't ask about my soil mix.
I didn't, did I?
Now, my typical soil mix is about a 1-to-1-to-1 peat moss, perlite, vermiculite. And more lately, since I've had trouble finding vermiculite, I've increased the perlite. I can get that more readily
Okay.
than I can the vermiculite. But drain, 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 and I always put a layer of perlite in the bottom of the pot for extra
I do
drainage.
that too. Okay.
So everything is really light and
Okay.
airy.
Okay, well that's great. That's really good to know, because my mix, I get a mix from Gary Makita.
Okay.
I use the Sunshine Mix,
Okay.
and that seems to work really well for
Mhm.
me. But I cut it again with
perlite.
more perlite.
Yep.
And I use a modified Texas style of
Okay.
potting, so
Mhm.
I definitely have that layer in
Yep.
the bottom.
Mm-hmm.
But I put a wick in all of my plants,
Mm-hmm.
but I have them, they're all individual saucer. They're
Yeah.
all individual watering. In the very beginning, I'll leave them in a community style tray.
Yeah. Mm-hmm.
But once once they're really growing, I kind of move them onto their own. But back to I wonder, can you? There are. I have a number of books. I'm stuttering now because I'm just trying to think in my head. There's so many things about these species. They're so cool. I have a book that I got from the UK that has like watercolor paintings of the species in it.
Yes.
I have
Mhm.
that. I mean, obviously the AVSA.
It's Joan Rothschild or something
Yes,
like
yes,
that.
and
yeah.
I just got it. I found it, and I thought, oh, I don't have that.
Yeah.
So I picked a copy up, I think, off of eBay. Of course, the AVSA handbook, which does talk about the species in it. I think there's another little teeny weeny book, and it had like the color things on the back and all kinds of stuff. There are a number of books about the species. Is there one, or are there one or two that you would recommend for people, or that you find to be valuable tools, or?
And I'm blanking out on the name. The Canadian lady who just published
Francine Poulin.
Francine's book is probably the most current and probably the best treatment of the species. And we consulted a great deal back and
It's
forth.
a great book.
On
We've
that one.
talked about it here on the podcast
yeah.
before. It's
Here's
wonderful.
just, as far as a single individual book on the species, probably that. You get lots of individual articles in the African Violet
Yes.
magazine from time to time. I had intended to write a book, and that fell apart. So a lot of my information went to Francine. So she's got a lot of the most current, recent,
Okay.
material
Great.
experiences
That's good to
there with that.
Very cool. Do you have a favorite species of
Oh,
all
geez.
these plants? You knew I was going to ask you that, didn't you?
Favorite.
Come on.
Oh goodness!
I mean, that's like that's like saying, do you have a favorite child?
Favorite child?
Right?
Yeah,
Okay.
it really is, and it's even worse on the species than it is for hybrids.
For hybrids.
For which of my hybrids? There are different things in the species that I admire them for, and so they'll take on their tone for that. The grandifolias are just wonderful plants to grow,
Okay.
just because they're easy, very floriferous. They shape well. They're just wonderful plants to grow. Velutina amazon, the tetraploid Velutina plant, is probably one of my favorites.
Now, is that different than a regular Velutina?
Yes.
Or a Velutina light? Or?
Yes, there's 3 different Velutinas. There's and I got all 3 downstairs. Velutina light, Velutina amazon, and the regular Velutina. Velutina light is a recent mutant that was discovered here in the United States, and it grows very compact. It's a mini, and has lighter blue flowers than the Velutina, but it's a very, very compact plant. Velutina amazon is probably twice the size of Velutina.
Maybe I could grow that one. I'm a standard grower.
Okay, it's absolutely huge. It's very coarse in that the foliage doesn't overlap particularly well for shape and for show. But the foliage is just as hairy as everything. You'd think Velutina is hairy. Velutina amazon is like another magnitude up
Oh, wow.
on hairy. Big coarse grower, throws up big tall spikes. They're darker blue-purple than what Velutina is.
Okay.
But it just a wonderful plant with that. Some of the trailers, if you're into trailing, the Pendula Kazari is a frequent one you see
I've
at
seen
shows.
that show a
And
lot, yeah.
it does wonderful for growing for show. Regular pendula has got nice foliage, but only throws up one bloom at a time. There's only one flower on that stalk, and so you can't ever get hardly any flowers on it to speak of. Grotziana is a wonderful looking plant, but a tremendous challenge to keep alive.
Is that the one you had?
And one of the ones
in
I had in the
domes?
domes.
Okay.
You wouldn't have to have it into the domes, but and I've never been able to get that one to bloom. I did manage to get Magangensis occidentalis to flower a couple of winters ago, and I was first time I'd flowered that thing. But it's another low temperature one that would do a nice windowsill plant. It's one of the nice things about the species. Some of them are cooler growers, and they make nice windowsill plants where other violets wouldn't survive.
So they can be closer to the windowsill.
Yes, they can be
and not-
closer to the
okay.
window. You know, streps are said to grow cooler than violets. And any place that you could get a strep to do well, some of these violets would do well. In fact, you could almost, if you wanted to, create almost an Arctic line of African violets. Ones designed for cooler growing conditions. And we've talked about that as a possible hybridizing goal if you wanted to get into that. But the temperature regimes of the wild plants are quite a bit wider than what the cultivars are. Ionantha, which is what most of the cultivars, hybrids, are developed out of, is a lowland plant species. It's a warm grower. And so is Confusa.
That makes sense.
And
then,
so
yeah.
it fits well with our domesticate, house plants kind of thing. But you've got some of the plants like Chimensis, which grows at 6,000 feet elevation.
It's cold.
It's colder. It doesn't freeze, but the nighttime temperatures drop down into the 40s. And so it does wonderful in a windowsill environment.
Great.
So you get these wider temperature tolerances in some of the species plants,
Okay.
which makes them challenging to grow if you try to put everything under one warm cultivar condition.
Well, you've given me a really good idea because I have this new shelf that hangs in the window, and I'm thinking that might be what I should maybe try to grow there.
Maybe some of the cooler
some
species.
of the cooler species.
Yeah.
But I mean, it's going to be bright, be probably an eastern or southern, maybe even exposure. I haven't decided which window yet. That's my only holdup.
Okay.
I haven't done it, but
Yeah, and the African violets are understory plants. So they're
understory plants. What does that mean?
understory means that they're the ones that you find on the forest floor underneath a tree canopy. So you never find the wild
okay.
African violets in full sunlight.
So
they're
they are going
always
to want
under
that south window. They're going to want.
They're-
Okay.
That's why they do well with filtered light, and why
Okay.
they do well in plant stands, which can't even hope to match the intensity of natural sunlight.
Okay.
And that's what you run into a problem with some of the other gesneriads, because some of them are full sunlight
Okay.
plants, which makes them difficult to grow indoors because of the brighter light conditions
They
that
just
they
don't
need.
get enough.
They just
Okay.
don't get enough.
Cool.
But violets are an understory, forest floor. Being from Illinois, you know that in the spring you get the wildflowers that bloom into the spring. That's the kind of environment the African violets are native to,
Okay.
underneath the tree canopies.
Okay. Okay. Very cool. Well, what advice would you give to someone who really has a developing interest in the species plants? How would you guide them? Where to start?
First off, finding them is going to be a bit tricky because there's not that many vendors who are carrying the species. And some of the rarer ones you're just probably going to have to find somebody who grows it and beg for a cutting.
Okay.
Because the cooks are no longer carrying them. And I don't know of anybody who's carrying the full range anymore, because it's a real challenge with that many different growing conditions
Sure.
to try to keep everything going. Understand that you're going to be growing them more for the plant in the foliage rather than the blooms.
Okay.
They're not nearly as floriferous as the cultivars have been bred to be. Not to say that you can't get good headed blooms in some of the species, because you certainly can. I've seen a well-grown Chimensis plant that had 70 to 80 open blooms on it at
Wow.
one time. But that's rare. I mean, you just don't see that kind of bloom count typically. They're going to be much more tolerant of differing temperatures, and you may have to experiment around a little bit to try to find the right area
Okay.
for them to fit in. And I would try cooler conditions
First.
and ends of shelves, a
Okay.
little lower light intensity, and just see how the plant is reacting, and then maybe move it, if you need to. Growing wise, treat them really a lot like your regular violets.
Okay.
Maybe a little less fertilizer because they're not going to be putting up the blooms, that kind of thing. And some of them will rarely bloom unless they get the temperature drop at night in order to trigger them. But a lot of the times, this is going to be more of a foliage plant rather than a flowering, per se. Not to say you can't get some that are known to be floriferous, more so. But these are going to enjoy more for the diversity of the plant form and the foliage style and the growth form rather than the flowers.
Awesome. Thank you. Thank you so much.
You're welcome.
Is so interesting.
I always love talking about the species. I'm really pro-species
just
guy.
Awesome. Well, and we should grow them. I think all of us as growers should at least try them because
and I would encourage
important.
Anybody watching this, keep 2 or 3. Try growing 2 or 3.
Yeah, start small.
Start small.
Limit your collection.
Okay. But keep 2 or 3 out there for the historical value, for the genetic value. But what I would really love to see is a network of growers out there, because if everybody grows 2 or 3 different ones, then instead of me having to jam 60 or 70 plants with replicates into one area, because I'm seriously, if we had a house fire, some of these things would be gone forever, because I'm the only one who has some of these things.
So I'm going home with some.
You're
right?
going home with some. You're going home with some. But that's what I need, and that's what we truly need. Is we need more people growing 2 or 3 or 4 of these, and then kind of a network or a register of who's growing what. Because if you have a meltdown, and it's easy to have something go wrong.
You're looking at that. I'm the poster child.
It's like when my collection crashed about 3 years ago, trying to find to replace these species. And I still haven't found everything yet. I'm still missing several things that I had before. And I don't really know whether they're still available. They may be extinct.
Could be gone.
They may be gone. I might have been the only one who had them. So, and I don't want to be that way. Perfectly happy to share material around. But keeping track of who's got what and where. And, Bill Price and I back each other up all the time.
Sure.
He has a crash. I send him stuff. I have a crash. He sends me stuff. We go back and forth, but there needs to be more than just 2 or 3 of us, doing that, really.
Okay. All right. Well, you guys, I'll be back in just a minute. Isn't he awesome? And when you guys, you did see his collection earlier this summer, he really is a living reservoir of the African violet species. His collection is so interesting, and just to hear him talk about it, I could listen to him all day long, as you guys know. So I hope that you enjoyed that, and I hope that you won't miss the look at what's on the stands too much this week. So it is time to get the bail money ready, and I do have one event that I want to share with you from the AVSA website. And actually, it's the last event that's listed there until February. So we're really at the tail end of the fall shows. And this is an international show. This is the Romanian Club of Gesneriaceae. I'm sure that's means Gesneriids, but I'm not pronouncing it correctly. But this is they are the Romanian Club of Gesneriids. I'm just going to say it that way. Forgive me, you guys, for not pronouncing it correctly. They are having their show in their African Violet and Gesneriid show from Thursday, November 14th to Sunday, November 17th. And this will be in conjunction with the Botanical Garden di Bradza Center of Ecological Education in Bucharest, Romania. So if you happen to be in Romania, or you happen to be traveling to Romania between the 14th and the 17th, and you're in Bucharest, I hope you will go and see the show and enjoy it. It sounds like it would be very exciting. It's a long show, 3 days. That's really cool. it's time to keep moving forward, you guys. This, like I said, has been a longer than normal episode. I've tried to kind of truncate it a little so that I left out the look, just so that we wouldn't run over an hour or too much over an hour. I mean, this is, I think, the longest episode we've ever done. But thank you so much for being with me this week, and I really hope you enjoyed this interview. As I said, I am traveling a little bit this weekend, and I hope to have another surprise for you next week. So you'll have to come back and see what that might be. Cross your fingers. I hope your days are filled with all the things you love. Good growing. I'll see you next time.